thank you guys so much for the opportunity to present our work. And it's a tough act to follow, Jesse and Betty, but I'll try my best. So we started on COVID pretty early on, and one of the first questions we wanted to ask are what host genes are actually essential for infection beyond just ACE2? All right, so I want to start by showing the people who actually did most of the work. So in my lab, Jin Wei and Mia Alfahara are two phenomenal postdocs. And then our close collaborators at the Broad, John Dench, and then Ruth Hanna and Peter DeWeert in John's group. So I'm going to first give you an overview of the CRISPR screen that we did. I'll then highlight some proviral and antiviral genes and pathways. I'll show you some data highlighting that we think these are potentially novel therapeutic targets. And then I'm going to focus more in on one of the top hits in the screen, a gene called HMGB1, and work we've done to try to elucidate the mechanism of its role in regulating SARS-CoV-2 infection. None of this is published. Some of it's already on bioRxiv, so you're welcome to check that out. So the premise of all of this is that we fundamentally believe that you need an increased understanding about how SARS-CoV-2 interacts with the host, both to develop better prognostic tools, therapeutics, including vaccines, and potentially drugs. And for instance, we don't really know why clinical presentation is so variable, ranging from asymptomatic to respiratory failure and death. Is this host variation? Is this obviously potentially differences in the virus or something else? And then, more immediately, can we actually develop host-directed therapeutics targeting host genes that are critical for infection? Obviously, drugs targeting ACE2 are one potential mechanism, but there are other potential unknown targets that we wanted to identify. So I just showed this SARS-CoV-2 life cycle briefly to highlight that the virus uses the receptor ACE2, and then it requires one of two proteases dependent upon the cellular context, either one called cathepsin L, which mediates proteolytic activation of the viral spike protein inside of the endosome, or another protease on the plasma membrane called TMPRSS2. And either of these two are sufficient, and they're expressed in different cells, and some cells have both of them. But beyond ACE2 and the proteolytic activity of the spike protein, essentially all of the other genes, the host genes that are essential for infection, are unknown. So we wanted to discover these in an unbiased way. So we did this by using a 4-genetic screen. We used a genome-wide CRISPR screen. And when we started, we screened a large panel of mammalian cell lines for susceptibility to SARS-CoV-2 infection. And we were looking for cells that endogenously expressed ACE2 because we wanted to find regulators of ACE2 expression, so didn't want to use an ACE2 overexpressing system. We wanted cells that we could infect the vast majority of the population, and we wanted cells that would succumb to virus-induced cell death to facilitate screening, tens or hundreds of millions of cells in a BSL-3 environment. And also by screening for cell death, it's really probably the last stage of our life cycle that we could easily screen for, as opposed to using a viral reporter, which would look at an earlier stage of the life cycle. So we used a cell line called Vero E6. So this is kind of the workhorse cell line of many virology labs. It's what most people use to grow the virus, including the inactivated SARS-CoV-2 vaccine that's currently in clinical trials in China. And the problem is that it's a monkey cell line. It's a monkey kidney epithelial cell line. And there was no genome-wide CRISPR library for monkey. So in collaboration with John Dench's group, we generated a Vero E6 specific CRISPR library to knock out all 20,000 monkey genes with 4 individual CRISPR guide RNAs 1,000 times each. We then took this large vat of cells and we challenged it with either a mock or a SARS-CoV-2 at different virus multiplicities of different MOIs, different cell densities, serum concentrations for different times. And then we sequenced the cells, the guide RNAs that were in the cells that survived infection. And this is just an overview of what we found. So on the left is a volcano plot showing genes that were positively or negatively selected in the screen. And we calculate a z-score because the CRISPR library includes 1,000 control guide RNAs that don't target anything. So you could use those in that distribution to generate a z-score. You see in the proviral direction, shown here on the right, the top hit is the receptor ACE2. You see cathepsin L is also down here as a positive hit. And then TMPRSS2 is not selected, which is a nice control because TMPRSS2 is not expressed in Vero cells. and it's known that entry in Vero cells is cathepsin L and ACE2 dependent. Beyond ACE2 and cathepsin L, nothing else that we found is known to be important for a coronavirus infection. The second hit in the screen is a catalytic subunit of a switch-SNF complex, which I'll talk about more in a little bit, called SMARCA4. And then in the negative direction, so these are antiviral genes, we found 3 of the 4 members of a histone regulatory complex called the HUCA complex. And here's just highlighted here A and cabin 1. Shown here on the right is just the distribution of all the CRISPR guides in the screen and the distribution of the individual guides for each of these top hits. So ACE2, SMARCA4, cathepsin L, and TMPRSS2. Okay. So then just to zoom in, instead of that 30,000-foot overview, to the top 25 and bottom 25 genes. So on the left are the genes that were essential for viral-induced cell death. So you see ACE2 is the top hit, followed by that SMARCA4. You see that each of these columns represents a different condition, which is either a different viral MOI, a different cell density, a different length of infection. And essentially none of that matters. Viral infection appears to be viral infection, and it's really nice because it suggests these host genes are essential and they're not really context dependent in the setting of the kind of an artificial tissue culture setting. What's also striking, if you just take a broad overview of these genes, is that other than ACE2 and cathepsin L, the vast majority of them are genes that function in the nucleus. And most of them are chromatin or histone modifiers, which was a bit surprising to us because we've done, I don't know, dozens of screen with other viruses. And with the exception of DNA viruses, for other RNA viruses, we rarely get genes that act inside the nucleus because these are viruses that largely complete the viral life cycle in the cytoplasm. So it limits the direct interaction with nuclear factors. But it suggested there's some really interesting biology to us. Okay, so we wanted to then validate the CRISPR screen. So we generated about 50 individual cell lines where we knocked out different hits that were either proviral, so those are shown in red, or antiviral in blue. And then we asked whether or not the virus could kill these cells. And that's controlled, it's relative to a control guide RNAs, which are in black. What you can see is that the vast majority of these resistance or proviral genes, when you knock them out, confer protection to virus-induced cell death. And then the antiviral genes, when you knock them out, they're more susceptible to cell death. So this is a nice validation. And if you just collapse those multiple guide RNAs down to at the gene level, and then you compare the original CRISPR screen, so this is the Z-score from the original CRISPR screen on the y-axis, where the relative cell viability of these 50 cell lines on the x-axis, you could see that the proviral genes are have increased cell viability with infection, and then the antiviral genes have decreased cell viability with infection, suggesting that this, the screen was robust and reproducible in an arrayed format, and it's not an artifact of the pooled system. Okay. Next, we did pathway analysis. So we wanted to take an unbiased view, and we looked at both positive genes that were both proviral, pathways that are both proviral and antiviral, and pathways that came up in both the proviral and antiviral direction. The most striking proviral pathway was the switch-SNF complex. You also got a TGF-beta pathway, viral translation pathway, and then some other nuclear pathways in the antiviral direction. So to zoom in on the switch-SNF complex, you see that essentially every gene in the complex was identified in the CRISPR screen in almost every condition. There's also a few genes that are not annotated in the pathway that are critical. They're also top hits. As far as the other pathways, we got this RUNX3 regulates CDKN1 transcription, and this was really driven by the enrichment of transcription factors Smad3 and Smad4, which are important in TGF-beta signaling pathway. We then got a cystatin and endosomal lumen pathway. And this is largely driven by cathepsin L. And then in some ways as a control, this pathway annotated as viral translation came up in both the positive and negative direction. And this is largely driven by elongation initiation factors. Okay, so what is the switch-SNF complex? So it is a highly conserved multi-subunit complex. It was originally discovered in yeast, and it's conserved throughout mammals and humans. Its function is to mobilize nucleosomes, so it can slide the nucleosomes and eject them from the DNA in the nucleus. it then exposes chromatin, and then it can regulate it can regulate transcription by opening or closing chromatin. It's interesting because it's mutated in about 20% of cancers. It is an ATP-dependent process, and the catalytic subunit of this large complex is the gene SMARCA4, which is the #2 hit in our CRISPR screen. We also got Smad3 and Smad4, which are these transcription factors that are important for TGF-beta signaling. And these are interesting because for SARS-CoV-1, it was shown that the virus nucleoprotein directly bound to Smad3, and that this regulated virus-induced apoptosis and potentially fibrosis, which is a long-term sequelae of SARS-CoV-1 infection, and potentially, in some cases, SARS-CoV-2. We didn't get any of the classic TGF-beta receptors, but we did get a TGF-beta superfamily receptor member called activin R1b. So we think that there's a link between activin R1b, Smad3, and Smad4, and apoptosis. So we wanted to ask, are there host-directed therapies that are already existing targeting these genes and pathways that we can leverage as potential therapeutics? So for both the switch-SNF complex and SMARCA4 in particular, and Smad3, there are existing small molecules. So these are called CIS3 and PFI3 respectively. And here we just treat drugs with a non-cytotoxic concentration of drug. We treat cells with a non-cytotoxic concentration of drugs and perform a viral growth curve. And you see between a 1 and 3 log reduction in viral replication when you inhibit either Smad3 or SMARCA4. Notably, these drugs are really lead compounds. They're not optimized, and we're currently testing more potent small molecule antagonists, which seem to increase this effect. This is true both in Vero cells and human cells. This is just shown a human cell line called HUH7.5. We've also tested another human lung cell line called KLO3. In all cases, we see between a 1 to 3 log effect at multiple time points with both of these drugs. So now I want to show you more recent data from a couple days ago to test whether or not these host genes that we identified in the SARS-CoV-2 genome-wide screen were important for other diverse coronaviruses and whether they're important more broadly in human lung cells. Okay, so the way we did this is we generated a custom CRISPR subpool. So we took the top 250 and bottom 250 genes from the original screen, and we used 10 CRISPR guide RNAs per gene to give us a lot of depth. And then we then sprinkled in a couple of other genes known to be important for coronaviruses, such as the MERS receptor called DPP4. And then we tested this a bunch against a number of different viruses. So we used SARS-2 again. We used a replication competent VSV SARS-2 spike protein. So this is from Sean Wilen's lab. We used the SARS-1 HKU5, which is a bat coronavirus recombinant. So it has the SARS-1 spike protein on this bat coronavirus. We then used 2 different MERS, a wild-type MERS and one with a tissue culture adaptation. And then we used influenza A virus, the Wisconsin strain, and then a coronavirus called EMCV. And if you just look at, take an unbiased clustering, what you see is that the 3 viruses that cluster are the SARS-1 recombinant, the SARS-2, and then the SARS-2 VSV recombinant. You see that next related are the MERS, and then really unrelated are influenza and EMCV. And to us, this suggests that the majority of hits for the screen are actually those that are regulating viral entry of both SARS-1 and SARS-2. A number of the hits are pan-coronavirus, or at least they target SARS-1, bat, MERS, and SARS-2. But really, they are not generic antiviral or proviral genes, as shown by the lack of correlation between the influenza and the EMCV survival. Just to zoom in a little bit, just to show you the MERS, wild-type MERS data versus the SARS-2, the top hit in the MERS screen is the receptor DPP4. The top hit in the SARS-2 screen is ACE2, and DPP4 does not come up in SARS-2, and ACE2 does not come up with MERS. But you see this nice positive correlation between the genes that are important for SARS-2 infection that are also true for MERS. We then wanted to ask, are these genes broadly true, or is it just true in Vero cells? And this was a bit challenging because we thought that a number of them would be regulators of ACE2 expression. So we used a human lung cell line called KLO3 that is actually quite difficult to work with. It only doubles every 8 days or so. So instead of doing a genome-wide screen, we did a subpool screen where we took about 25 genes, both in the proviral, antiviral direction, and controls, and then we tested whether or not these genes conferred resistance to SARS-CoV-2. So once again, ACE2 becomes the top hit in the KLO3 subpool. We don't actually get cathepsin-L, which is not essential in KLO3s, but we do get TMPRSS2, which is essential in KLO3s, but not Vero cells. And then we get validation of the switch-SNF complex. Several of the other top hits in the Vero screen don't validate. And then we see some of the antiviral genes that are true in both settings. So we think that the Vero cell screen is highly predictive of what we would see in a genome-wide KLO3 screen. All right, so next I want to focus in more on a specific hit that was really of interest to us called HMGB1 and what the mechanism of action is in promoting SARS-CoV-2 infection. So HMGB1, it's a high mobility group box 1 protein, and it's really a pleiotropic protein. You can look it up on PubMed and find a variety of different functions. So its main function is to be a nuclear DNA binding protein where it can regulate transcription. Under conditions of stress or potentially viral infection, it will translocate from the nucleus to the cytosol and can even get secreted into the extracellular environment. In the extracellular space, it can function as a chemokine. It can directly bind LPS and potentiate the activity of LPS. It even has a receptor called RAGE that leads to NF-kappa B upregulation. And it's been really implicated in a variety of inflammatory diseases, most notably sepsis. So it's broadly thought to be a pro-inflammatory molecule. Okay. So the first thing we did was validate HMGB1. So we made individual knockout cell lines for this. We used 3 different CRISPR guide RNAs that gave us varying degrees of knockout. And you could see that the cell survival in response to wild-type SARS-CoV-2 infection correlates nicely with the original disruption by Western blot. So where the green and the red have better disruption, they have better protection. And if you look at viral replication, it also correlates in the same direction. So we see about a 2-log decrease in viral replication when you knock out HMGB1 at the bulk level. These are not complete knockouts, so they're not single-cell clones. Okay, so we didn't really have a great sense of what HMGB1 could be doing kind of a priori. So we just did what everyone would do, which is we just did RNA-seq of the HMGB1 knockout cells. And shown on the y-axis are genes that are upregulated in the HMGB1 knockout. And down at the bottom are genes that are downregulated in the HMGB1 knockout. And then on the x-axis is the z-score from our original genome-wide CRISPR screen with SARS-CoV-2. So what we were anticipating is or hoping rather, is that we would find genes in this top left quadrant, which would be upregulated in the HMGB1 knockout and identified as antiviral in the original CRISPR screen, or genes in this bottom right quadrant, which would be proviral in the CRISPR screen and downregulated in the knockout. And much to our pleasant surprise, we said no genes in the top left quadrant that met our predefined cutoffs, and in the bottom right quadrant, we only got 3 genes. So HMGB1 itself, which is a nice control. Then we got ACE2 and cathepsin-L, which are really, of the 20,000 genes in the genome, the only genes in which we could really identify a function for. So it made downstream studies straightforward. So this suggests that HMGB1 is regulating ACE2 RNA expression. So we wanted to look more closely at this. So using parental control cell lines or 2 different HMGB1 knockout cells made by CRISPR, this is just the plots of RNA-seq where each green spike is the number of reads, and there are fewer transcripts in the HMGB1 knockout, consistent with that last plot I showed you. We then, in collaboration with Will Culligan, Matt Simon, and Xin Yan's lab in West Kai, we did H3K27 acetyl chip-seq. So this is a marker of active enhancer elements. So this is the ACE2 locus. And what you can see is there's just a statistically significant reduction in H3K27 acetyl at the ACE2 enhancer element. Also, with Matt and Shin's lab, we did ATAC-seq, looking at chromatin accessibility at the ACE2 locus, and we see a trend towards a reduction in chromatin accessibility at ACE2, suggesting that HMGB1 is regulating both the enhancer expression and the chromatin accessibility at the ACE2 locus. So more recently, we generated single-cell knockout clones of HMGB1 and then complemented them with a functional wild-type human HMGB1 with lentivirus. And then we used a pseudovirus assay. So this is a VSV that expresses luciferase, and instead of the VSV glycoprotein, it expresses the SARS-2 spike protein. We also utilized the SARS-1 pseudovirus, an NL63 pseudovirus, and a MERS pseudovirus. And the SARS-1, NL63, and MERS pseudoviruses are all on an HIV core. And all this data is normalized to a VSVG. So what you can see for SARS-2, which is shown in red, in the wild-type cells, you get nice pseudovirus infection, and the knockout, you get a significant reduction, and then you can rescue that by complementing with HMGB1. And you see the same trend with the SARS-1 and NL63. And it's important to note that SARS-1, SARS-2, and NL63, which is the seasonal coronavirus, all use ACE2 as the receptor. And as a nice control, we use MERS pseudovirus. So the MERS uses DPP4, but not ACE2, and we see no significant reduction in either the knockout or the complement with MERS, really consistent with HMGB1 regulating ACE2 expression, which confers resistance to ACE2-dependent entry of coronaviruses. So to tie this together, this is just a model of many of the hits, and this is very much speculative, so if anyone has ideas, I'd love to chat more. So everything on here was a hit in the screen. So SARS-CoV-2 binds to the receptor ACE2, which we identified. It uses cathepsin L in Vero cells to finish viral entry. HMGB1 regulates ACE2 and potentially cathepsin L expression. We identified this TGF-beta superfamily receptor and the transcription factor Smad3 and Smad4. So we think that Smad3 and Smad4 may heterodimerize and enter the nucleus upon infection, and that this may direct the switch-SNF complex, which is this chromatin regulating complex, to turn on a pro-viral gene expression program. We think this potential pro-viral gene expression program is also regulated in the positive direction by other histone modifiers that we identified in the screen, and then it's antagonized by this hookah complex, of which we identified 3 members.
You have 2 minutes if you can start wrapping up.
Yep. ASF1A and HERA as antiviral genes. And work in the lab is really focusing on the mechanism of these other genes. So to wrap up, we performed a genome-wide CRISPR screen in the monkey cell line Vero E6 cells, which identified ACE2 and cathepsin L as critical pro-viral molecules, but it did not identify TMPRSS2. The vast majority of other genes identified in the screen were involved in histone and chromatin regulation, and this is both in the pro-viral and antiviral direction. Most of the hits were broadly active against other pandemic coronaviruses, including the SARS-1 bat coronavirus and MERS, and not other viruses. And then actually most of the hits mapped to viral entry, but not all. Using a subpool, we showed that using Vero cells was predictive of what we would find in a human lung cell line called KLO3, suggesting this isn't just a Vero cell specific phenomenon. And that small molecules targeting several members of the several hits from the screen, including Smad3 and the switch-SNF complex, can actually inhibit viral replication and may represent lead compounds for host-directed therapeutics against both SARS-CoV-2 and other potential future pandemic coronaviruses. And finally, we show that HMGB1 is a novel regulator of ACE2 expression in multiple cell lines in vitro. So just to thank the people who did the work. So everyone in my lab contributed in some way, but is really spearheaded by Jin Wei, Shin Yan's lab, Matt Simon, David Van Dyke, and the Iwasaki lab were also great collaborators. John Dench at the Broad and his group, who's been a long-term collaborator of ours, and really been wonderful to work with them. And then, of course, others who have provided reagents and expertise and funding sources.
Thank you, Greg, for a great talk, very stimulating. Did you find other classes of genes that are known to be downstream of TGF-beta receptor that were activated?
Yeah, so no, not really. Nothing that was, I would consider robust. So we don't know, it seems to be just Smad3 and Smad4, but we're not really getting a classical Smad3, Smad4 response. What we think is that, so the switch-SNF complex, which regulates chromatin, doesn't actually have DNA sequence specificity. So the way it targets specific DNA sequences is binding to other molecules that can bind DNA. So the hypothesis is that the switch-NIF complex is binding to SMAD3, SMAD4, and SMAD3, SMAD4 is directing the switch-NIF complex to SMAD3 target sites.
Other questions for Craig?
This is Eric. If I could ask a question.
Yeah, please.
Craig, I know that Nevin Krogan and company out at UCSF have been mapping protein-protein interactions between SARS-CoV-2 proteins and host proteins. And I'm wondering, is there any overlap between the guys you're finding in your CRISPR screen and this sort of physical protein-protein interaction map that Nevin Krogan and company have been generating?
Yeah, so obviously one of the first things we did when we got the results for all these screens is we compared ours to the protein interactome, both the original one from Nevin in Nature and then more recently the phosphoproteome, and in the positive direction. So of proviral genes, there is zero overlap between them. In the antiviral genes, there are two, AP2M1 and another, but the magnitude of the effect is very low. So I think it really highlights that a functional 4-genetic screen provides additional information beyond what you would reveal with a protein interactome, which is just looking at obviously direct interactions. In the case of the first interactome paper of just overexpressed proteins in 293 T cells.